Today we will discuss about the transient changes in the skin bacterial diversity when they are treated with UVB or cis UCA, when they are either treated with cis UCA or exposed to UVB. The data are collected at 8 hours and 24 hours, and 8 hours is considered as a base level, and with 24 hours they can see whether the changes are increased, decreased, or remain same levels. So once they collected their samples, they did this treatment, monoazide treatment, to get rid of the noise, we can say that. And after that they did the DNA isolations and do this 16-hour RNA sequencing, and thereafter they analyzed this alpha diversity, beta diversity, taxonomic analysis, and shared features in order to find out what are the changes occurs in the skin bacteria because of cis-UCA or UVB. And what they are able to see that in case of this alpha diversity, they see a significant changes after 8 hours. So baseline is not 8 hours, I have mistaken that. So they check the baseline is before the treatment. So compared to the baseline, they can see a decrease in this the diversity Shannon's diversity index for cis-UCA treated mice and also for the UVB exposed mice. And when they check the beta diversity, here also they are able to see that the changes are quite significant when they compare it for this 8 hours, and they can see that two different clusters are formed with R value for this UCA cis-UCA treated mice is around 0.39 and p value is 0.014, and whereas for the UVB exposed mice we can see the R value is 0.23, indicating that for this cis-UCA treated mice models we can see more diversity compared to the UVB exposed mice models. And when they check these signals, when they compare the signals of the baseline with the 24-hour signals, they are able to see that for the 24 hours the changes, the Shannon diversity index for this cis-UCA treated mice models is again reached to the baseline levels, and the difference is non-significant. But for the UVB exposed mice models, the difference is statistically significant, although it reached close to the baseline levels, but the difference is still statistically significant. And when they check the beta diversity, here also we can see that for the case of cis-UCA mice models, we can see the R value decreases from 0.39 to 0.17, and also p values is greater than p value is we can say 0.136, and usually p value needs to be 0.05, less than 0.05 in order to be statistically significant. So in this case also we can see that for the case of cis-UCA treated mice models, the diversity goes down after or we can say that the transient changes in the skin bacteria occurs, and it is reversible in the cis-UCA treated mice models. But when we checked it for the UVB exposed mice models, the changes seems to be non-reversibles, or changes seems to be consistent even after 24 hours, as R value is 0.48 and p value is 0.006. So this indicates that the effect of cis-UCA is more reversibles and can be seen in the 28 hours time period, whereas for the case of UVB exposed mice models, the results are more consistent even even after 24 hours of being exposed to UVB. So these are the some of the transient changes, preliminary informations that they have presented in their first figure. If you need to understand more about this transient changes in skin bacterial diversities in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.